What is the genetic engineering?
Genetic engineering is the direct manipulation of an organism's genes using biotechnology. It covers different kinds of technologies used to alter the genomes, includes the insertion of genes from other individuals, either the same or from different species, that aims to produce or improve products.
Genetic engineering can be accomplished using multiple techniques. There are a number of steps that are followed before a genetically modified organisms or GMO is created. Genetic engineers must first choose what gene they wish to insert, modify, or delete. The gene must then be isolated and incorporated along with other genetic elements into a suitable vector. This vector is then used to insert the gene into the host genome, creating a transgenic or edited organism. The ability to genetically engineer organisms is built on years of research and discovery on how genes function and how we can manipulate them. Important advances included the discovery of restriction enzymes and DNA ligases in the development of polymerase chain reaction and sequencing. This allowed the gene of interest to be isolated and then incorporated into a vector. Often, a promoter and terminator region was added as well as a selectable marker gene. The gene may be modified further at this point to make it express more efficiently. This vector is then inserted into the host organism's genome. For animals, the gene is typically inserted into embryonic stem cells, while in plants, it can be inserted into any tissue that can be cultured into a fully developed plant. What is the role of genetic engineering in our life or in human life?
In medicine, genetic engineering has been used to mass-produce insulin, human growth hormones, Foley stem for treating infertility, human albumin, monoclonal antibodies, anti-hemophilic factors, vaccines, and many other drugs. In research, organisms are genetically engineered to discover the function of certain genes.
What are the roles of the DNA, genes, and protein in a given trait?
DNA carries the instruction to produce proteins and it's responsible for passing traits from one generation to the next. How would you relate the individual trait or characteristic to proteins and gene in DNA? Traits are determined by genes and also they are determined by the interaction with the environment with genes. Remember that genes are the message in DNA that define individual characteristics, so that the traits is the manifestation of the product of gene that is coded by the DNA. Would the manifestation of trait be affected once the DNA nor the genes are altered? Yes, it is affected because DNA is a dynamic and adaptable molecules. It is subject to change as a result that called mutation.
What is the recombinant DNA technology?
Recombinant DNA technology, it is a technique used in genetic engineering and joining together of DNA molecules from two different species. For example, plant with new trait. We will use agrobacterium method to form a plant with a new trait. First, agrobacterium tumefaciens will cultivate with plant pieces. When we say agrobacterium tumefaciens, it is a soil bacterium that has the ability to infect plant cells and transfer a defined sequence of their DNA. It contains a T plasmid that is carrying desired genes. When agrobacterium and plant pieces put at the same place, that is the time where DNA coming from agrobacterium will be transferred to the plant cells. In the plant cells, we can now see the chromosomes with integrated DNA that has encoding desired genes. After that, cell multiplication or callus will happen. When we say callus, it is an unorganized plant parenchyma cell, and followed by generation of root and shoot of the plant with new trait.